So good afternoon. My name is Yann Herault. I'm working in France, and I've been studying DYRK1A for many years in a different perspective at first. Turn on your... Is it better now? Yes. Okay. Good. So start again. I'm Yann Herault. I'm a senior director of research in France. I've been working on DYRK1A for many years, mostly because I was interested in Down syndrome, and we have shown that this is a very important target for some of the neurocognitive disorder and neurocognitive defect that is appearing in those people with Down syndrome. So I'm based here in Strasbourg, in the eastern part of France, close to Germany. It's just at the border, in fact. And I'm working in this institute. This is a building which has been made by a US architect. And I'm directing, in fact, this part of the institute, which is called the Mars Clinical Institute. This is a beautiful city. If you have a chance to come, let me know, because it's very nice to visit. And this Mars Clinical Institute is really devoted to make models for rare disease. We have more than, I don't remember, more than 100 or 200 models, including loss of function or specific mutation for rare disease. And I'm interested in many of those. And this is made by strong geneticists, and we have physiologists, behaviorists, and also we are doing a lot of preclinical studies to have proof of concept in order to go further into demonstrating the validity of that. And this is what we aim at with our models. And we work with either mouse or rat models, first to understand the physiology and the function of the brain, but also the gene function. We are involved in this international mouse phenotyping consortium that I was sharing for many years. We are also trying to understand the variation in the genome, which are really causing a disease or maybe promoting some disease due to also interaction with the environment. And we are also developing those preclinical studies in order to have proof of concept. Saying that, so DYRK1A, of course, we heard about the mini brain story, which is very nice, because without that, I think it would have not been raised as a candidate for Down syndrome. And this is why it has been very carefully studied in Down syndrome, because this is located in the human chromosome 21, and it is also found in the regions which are homologous in mouse to human chromosome 21. And when it is overexpressed alone, it is able to induce many different cognitive defects in those mice and in other models. And we have now a series of inhibitors that allow us to reduce the kinase activity, which is in fact the kinase function of DYRK1A, allowing to reduce some of the phenotype. And I will show you that. And this could be of interest because if you remember the paper by the group of Herault, it's a group of very talented people in Barcelona. They showed that over 26 mutations that they scanned, in fact, many of those were loss of function for DYRK1A syndrome, but a few, 5 of those, were showing higher level of DYRK1A kinase activity. So maybe we have here, we might maybe consider that if we want to go for personalized DYRK1A syndrome treatment, because this maybe, we maybe have some clue here. If we take into consideration, for example, the effort we have done with working on Down syndrome, where we have 3 copies of DYRK1A and another activity of DYRK1A, I have no, I will show you something maybe. This is what we can obtain. So this is a new series of inhibitors which has been developed now with a company in Brittany, which is called Pera Pharmaceuticals. I'm working with them for years now. We start with another inhibitor which was called leucetine 41, but now we have this leucetinib. This is quite complicated, but we can just focus on this part. Here you have wild-type animals which has been non-treated or treated with these inhibitors. They are still able to make a very simple test, which is to spend more time looking at a new object compared to a familiar one. This is a normal curiosity of the mice. They go for the new object rather than the familiar one in this test. Okay? So this is what they do when they are wild-type. They go for the novel object versus the familiar one. And if you treat them, there is no change, which is good, in fact, for the drug. And now, if you look at the transgenic for DYRK1A, they are unable to make the difference between the novel object and the familiar object. They explore the same amount of time. And if you do the treatment, you can restore this activity. Having the mice treated, the transgenic mice, which are only overexpressing 1 copy of DYRK1A, able to recover the capabilities of restoring this type of memories. differentiating the novel versus the familiar one. And we can use different systems. I have no time to discuss that. But what is also very interesting is that if we look now at the phosphorylation of one of the best candidates for phosphorylation activity of DYRK1A, which is a tau at a very precise serine position, you can see that there is an over increase of phosphorylation of tau compared to the total amount of tau in the transgenic for DYRK1A. And when we treat with a drug, which is called the lecithin 10-ES, but we have much more, you can reduce the activity of phosphorylation of DYRK1A, showing that we can really target the kinase with this drug. So the DYRK1A kinase. So there is maybe something to think about here for those gain of function. Saying that, is it true? I think it is. So we have made a lot of models for DYRK1A. I just put a few here. We have a complete deletion of the gene. We have a conditional knockout. I will show you how we use it. We have different point mutation. We work with Jeremy on the T588N, which was a variant of unknown significance. And unfortunately, it's not really a very strong mutation of DYRK1A. In heterozygote, there is no phenotype. When you start to make it to the homozygote level, you see something, you see some defect. But it's not what we are hoping for a normal, let's say, haploinsufficiency effect. The kinase dead is very strong, and it's mimic most of the mutations which are affecting the kinase activity of DYRK1A. It's a very specific mutation, the K188R, and if you look at it as a kinase activity in heterozygote, it's almost 50% of the wild type level. But you can also appreciate the variation of the kinase activity in standard conditions, which is something that we still have to explore. And this is the transgenic for DYRK1A that I have no time to show you for the effect of the lecithin, but I have it in my computer if you want, it's somewhere else. And what is really striking here, we made this mutation, which is called, so this is not published, so please don't put that onto the network, but this is the A195T mutation, which has been shown by Herault to be an overactivity of DYRK1A in vitro system. And in the mouse, when we do this mutation, we have indeed a larger increase of DYRK1A because now you are running between 4 to 5, to around 5 times the level of expression, so 2.5 the time of the level of expression of the wild type. So this is what we have. So maybe for those, I would be curious to see if we can treat them with an inhibitor of DYRK1A and restore some of the function. So we are doing that at the moment, not the treatment, just the phenotyping of those mice to see how far they are affected by the overexpression of DYRK1A. But they should be at least similar to the transgenics, maybe more. So this is something that we are doing. And we have also another, the zoo. We have been sophisticating the zoo for DYRK1A mutants by having now rats, a model with carrying a deletion, and we have 2 duplications of the region of DYRK1A. The first one that we did was quite interesting, but has no phenotype at all. And indeed, there is no, even if there is 2 copies of DYRK1A, there is no overexpression of DYRK1A. We had to make a duplication from the last region of the previous gene to the start of the next gene to get an overexpression of DYRK1A and to get phenotype associated. I have no slide about that, but if we can show that later on. When I make a set of slides, I had it, but I think it's no more valued now because there was already a very nice talk about the role of DYRK1A in Drosophila. But I think what Paco Tejedor has shown, and also the previous speaker, is that there is a strong effect of DYRK1A during the proliferation of the progenitors. This is what he has been focusing on. But there is also some effect of DYRK1A during the neurogenesis, and during also the maturation of the neurons, and also during the dendritogenesis, which is when the neurons are making contact with the others. And we think that, based on our experience, and I have no time to discuss that today, it has also a very strong effect on the synaptogenesis, because we find, for example, that DYRK1A is able to phosphorylate one of the key genes, which is controlling, I think, the pool of vesicles which are secreted to activate the next neuron, which is called the synapsin 1. So it's in a previous paper. I can show you the reference. But what I would like to emphasize our work today on is about the brain development. So we are trying to address these major brain development processes using rat and mouse model. And the idea is that you have a kind of very good parallel, in fact, between the timing of the different, let's say, neurogenesis, synaptogenesis, synaptic pruning, and so on, that you can detect in the rat or in the mouse. It's a mouse a bit earlier, and compared to human. So which is something very interesting, because we could, of course, parallel the study to what we could expect to obtain in human. As you may know, in the brain, there are two main types of neurons. There are the excitatory neurons that we call also the glutamatergic neurons. They are from different types, there are different types and different kinds. They are located in different places of the brain, but most of them are in the cortical region. And they correspond to about 80% of the neurons in the brain. And there is a lower subpopulation, which is called the interneurons or inhibitory neurons, which are also very diverse, depending on their localization in different cortical regions, for example. And they make contacts with the glutamatergic neuron, and their role is really to control the signaling between neurons. So they are more like let's say, slowing down, for example, the activity of the other neurons. That's a man. Or there could be a double negative loop also, but I don't want to go too much into detail for that. But so those ones are really the regulatory neurons that control, in fact, the activity of those ones which are the excitatory neurons. So if you have too much of that, what is the main problem is epilepsy, for example. Okay, this has been shown in many different cases. So the balance between interneurons and excitatory neurons is really key, for example, for controlling epilepsy. And I've been working on another gene which is in fact controlling the maturation of those interneurons, which is called IRIX, and involved also in epilepsy, is really striking for that. But still, so if you look at DYRK1A, and those neurons, they are not born exactly at the same place. Yes, let's put it in the right correction. In the right, I don't have more. Okay, I think I don't have more. So they are not different. So the cortical do you have somebody that's a pointer or not? Yeah. Oh, you are.
It's very similar. I think there is the plug here. So let's take this out.
Yes.
Put this in.
Oh, thank you so much.
See if it works.
Yes. Okay. Thank you so much. So the excitatory neurons and the inhibitory neurons they are not going to be born at the same place during development. So this is a section of a mouse brain. It's a bit artificial, but let's put it this way. And here you have the dorsal part of the brain, the ventral part. And here you are cutting the brain in the middle, around the middle. Okay, so this is about 13.5 days of development in the mouse. And the cortical and excitatory neurons, in fact, are born from a layer of cells which are located here, which are just above what we call the ventricle, which is an empty space, in fact. And there is a lot of good stories on the role of DYRK1A in this population of neurons that I have no time to tackle today. And the inhibitory neurons are born here in what we call the ganglionic eminence. And depending on the position from the rostral to the caudal region, they are going to give birth to different types of interneurons. It's very complicated, I must say. I don't understand all the things here, and they are called those ganglionic eminence, LGE, AGE, and CGE, but CGE is for caudal, this is the lateral, and this is the medial part. And some of them will stay also at the same place, in the place which is called the striatum, which is very important for controlling five movement. We have a story which is running on that. I have no time. I cannot disclose it today, but it's very interesting for precise movement. But today I'm going to focus on the role of DYRK1A in those two types of neurons and what we did for that, because DYRK1A is indeed expressed in the excitatory region, in the ventricular zone where the progenitors are. So it's probably similar to what you observe in Drosophila, but it's not the same organization in mammals. And here you have also expression of DYRK1A, which has been detected. I think it's a previous works from Amerle here in the ganglionic eminence, and this is a very specific map. If you want, I can comment it, but this is the level of expression of DYRK1A along the level of expression of DYRK1A along time, and you can see that it changes from tissue to tissue and cell type. It's very sophisticated, but it's very dynamic. Let's put it this way. What we did first is with Véronique, a colleague of mine working in my lab, which is a senior scientist. We did the inactivation of DYRK1A using the conditional knockout. It was quite complicated because we did that also for Down syndrome and also for MRD7, so which is also old name for the haploinsufficiency syndrome due to DYRK1A. We did the inactivation of DYRK1A only in the cortical region, only the glutamatergic red neuron using genetic tools, let's say. You use the conditional allele with the driver of the tree, for those which are known, who know about this system, expressing the tree under the CAMK2, which is a very specific enzyme from the excitatory neurons. And with that, we were able to detect that, for example, the DYRK1A loss, one copy of DYRK1A was causing a loss in terms of recognition of the novel object in DYRK1A, in the DYRK1A CAMK2 model. So where only DYRK1A has been removed in the glutamatergic neurons. So this means that DYRK1A dosage is very important for this type of memory in the cortical region. Okay. For the social preference, we have no change. I'm not going to comment the fact in the Down syndrome model, but we could if you want. And we find out that there was a commonalities between what is observed in Down syndrome model and the loss of DYRK1A in this model. But this has been published, so you could, if you want, you could go back to the publication. What I would like to introduce you is a really recent publication. It just went out beginning of July. And this is the work of 3 talented people in my lab, 2 postdocs, Maria Victoria Inkeman-Rivas, Aline Dubos, and Victorine, a new PhD student. She will be finishing around beginning of next year. And they demonstrated that, in fact, DYRK1A haploinsufficiency is really key for interneuronal migration, which is another step of their life. I will show you what it is. During corticogenesis, and this induces a series of defects which are really relevant for the DYRK1A syndrome. Oh, we did that. It's genetics. We have very powerful, so we have not such same of tool as that were shown before in Drosophila, but we have other tools, so we can label also interneurons using this GFP marker, which is expressed now under the GAD65 promoter, which is specific for the series of a large series of interneurons. We bring here the conditional allele of DYRK1A, which is located between these 2 arrows here. And we bring here another pre-expressing line, which is now specific for the interneurons. But this is so specific that it is only expressed after the proliferation of the progenitor of interneurons. Okay, because we don't want to have to mess up the proliferation here. It could be done. This could be done with another strategy. But the idea was to bring everything together to get an inactivation of DYRK1A, of one copy of DYRK1A, only in interneurons, and to compare with animals in which we won't have this conditional allele. Okay? So you have the DYRK1A DLX, we call them, and the DYRK1A of the control population. Let's put it this way. And what we are working on is that if you look now at the migration of interneurons during development, because those neurons, they are born here, but they have to migrate into the cortical region. And they do that very actively. It's a migration process. They are taking 3 mainstream. We'll show you that maybe later on. I think the picture are a bit overburned here, but I think you can see somehow. So these are different positions of the section from the rostral part to the caudal region, so going through the different ganglionic elements. And you can maybe appreciate, so this is the control. You can see that you can detect GFP cells, so they are not green here, but they are black, right? So it doesn't matter. You can see many cells here coming from the ganglionic elements. These are interneurons which are migrating into the brain to reach the cortical region where they should be at the end. Whereas here you have a very few. And if we go to the caudal position, it's exactly the same. We have a very few number of cells which are migrating, while in the controls we have some. It's not that much, huh? It's not that much cells, but it still makes a difference. So if you look at numbers now, if we count the number of cells which are migrating, there are really more in the controls than in the DLX DYRK1A inactivation in every region that we have been looking at. And we did that on several embryos, of course. We did even a more sophisticated analysis in which we look at the flow of migrating cells. So we can cut, for example, here the different sections by different, let's say, a band of the same size, and we look at the number of cells at each position, and you can see, for example, that there are much cells which are progressing into the dorsal part of the brain. The dorsal will be here, compared to the D-helix inactivation of DYRK1A. So it seems that without having the normal dosage of DYRK1A in those interneurons, we are not allowing them to migrate correctly in the brain. And what happens later on at P8, so after birth, 8 days after birth, there is a critical period for interneurons development at P7. So this is what we did at P8. We still see the same. So those cells are less in the cortical region. So these are different types of interneurons. There are somatostatin, cal-binding, GAD65 positive. So these are the whole population. And these are the cal-binding ones. So these are different kinds of interneurons. They will make contact with different glutamatergic neurons at different positions. But you can see that for all of those populations, there are less neurons that we can score in the mutant compared to the wild type. Last year for the cal-binding. And if you look at their distribution along the dorsoventral axis, it's completely messy. So you should have more neurons like the wild type here, whereas here you can find more neurons in the more ventral part. So it seems that it's like if you have still a migration, but which is impaired, and they never reach, in fact, they seem to never reach the correct position at the end. It's really important because in terms of physiopathology, it's maybe more complicated. And when we did the similar experiments, so later on in adult mouse brain, we see the similar differences. Most of the neurons are not well placed in those mutants. Having one copy of DYRK1A less in those interneurons during their migration step is really reducing their capabilities of migrating and to incorporate into the different layers of the cortical region. Okay. So this is what I summarize here. The haploinsufficiency of DYRK1A leads to a decreased number of interneurons in the cortex. This is for sure. We haven't found any evidence of cell death of proliferation deficit, but this is what we were looking for, and cell death could have been an option. Huh? There is a delay in migration fronts, so they seem to migrate, but they don't migrate very efficiently. And there is a decrease of an abnormal localization of interneurons at P8 and also in the adult later on. So which is a bit of a mess, because if you want to change that, that will be quite complicated, I must say. But so what we did to confirm that, we went into an in vitro system, so we can do that. So using the same GAD-GFP, which labels the interneurons in the ganglionic eminence, we can take the ganglionic eminence from embryos, put them into culture, and follow now the interneurons which are migrating with different trajectories. It's not well organized here, I must say, but still you can look at, I don't know if you can follow the track here, but this is one migration, for example. Another one has been through this way. Another one has made this route, and so on. And of course, we don't look at the ganglionic eminence because it's too fluorescent, but we look around. And we can follow, for example, the progression of the movement of one interneuron in a dish. And you can appreciate here, we can follow it, it makes this direction. And here, this is a mutant interneuron. And you can also appreciate that it makes it much more slowly. This is the same time scale. And indeed, and even the way it progressed to migrate is different. So there are different details that I have no time to discuss, but it's quite interesting in terms of cellular biology. And the mean velocity overall of the migration of the speed for movement is reduced, and there is also an increasing time because it's not linear. In fact, the neuron when they move. Maybe I have a drawing somewhere. Yes, it's here. When they move, they make some projection. So they try to reach something. Then certainly they find their way, and then they move their nucleus. So there is another step which is called the nucleokinesis. So the nucleus of the neurons, where the DNA is located, is then moving one round, and then it starts again. You have a new elongation here, and then you have the movement of the nucleus, which is called the nucleokinesis. And when we look at the nucleokinesis, it's reduced, in fact, in the DYRK1A mutation in the DYRK1A mutant. So this is really a cell autonomous defect that we are observing here due to the only one loss, one copy missing of DYRK1A. And this is also the amplitude, so the size of the movement is also reduced. And we did that. So there are also other chances that I have no time to. So the number of processes that are there sending around is much more complex when you have reduced dosage of DYRK1A in those interneurons. So in terms of, yes, sorry, did I miss it? Yeah. So in terms of conclusion for this part, we demonstrated that there is a slower migrating interneurons also in the dish. This is due to a less frequent nuclear translocation, a defect in the neuron length, which is shorter, and also an increased complexity of secondary neurites, which are projection of the neurons to reach new place. Let's put it this way. What we tried to demonstrate later on is that we were thinking that is it due to the dosage of DYRK1A or to the loss of the kinase activity? Because we know that the kinase activity can modify other proteins by phosphorylation, in fact. And we did that. We used a kinase mutant. We have a kinase dead mutant, so it's not able to make any phosphorylation, but the protein is still produced. And here we also look for different type of interaction with the drug, with a kinase inhibitor. And this is what Vicky did, in fact. So if we use the kinase dead mutant, you can appreciate that, for example, so we did, we verified that there was a decrease of DYRK1A kinase activity in the kinase dead mutant in this region of the brain. And, you know, you always have to control this type of things. And then when we look at the number of interneurons which were migrating in this K188R mutant, heterozygote mutant, you can see that there are lower number of interneurons which are migrating in the rostral part, in the medial part, in the caudal part, compared to the control. So it's really a kinase-dependent mechanisms which is affecting here. And yes, so I don't know, I don't think that I have time to go maybe too much into details, but we have been investigated what could be cellular and molecular mechanisms here. There are several which could be affected because we have heard that DYRK1A is also interacting with microtubule, which is indeed a very strong interaction. But still, it's also it has a role also in controlling the nucleation, the branching by phosphorylating NWAS, for example. And we were wondering if it could also have an impact on the formation of actin filaments, which are really important for making this process that the neuron use to project themselves into during the migration. And what we found, that indeed there is a change in the phosphorylation of cofilin, which is a very important protein for making the filiary component. But still, we don't see a major in the soluble or the filiary actin in those brain extracts. But what was really interesting is that there is another mechanism which is in fact very important for the movement of the nucleus, which is due to some actomyosin networks which are now built on the top of the actin filaments, and this will make the nucleus to move. This is micromotors, in fact, and this is controlled by the phosphorylation of with this myosin light chain kinase, and we find out that this phosphorylation is indeed perturbed in this mutant. So it seems to be one of the reasons why this nuclei is not moving at the same speed and with the same effect. Saying that, now, okay, I lost one slide. Sorry, I went too far. So we try to do the same type of experiment using an inhibitor of DYRK1A. So I show you that we have good inhibitors for the kinase activity of DYRK1A. And if we now treat with another inhibitor, which is called LCTD21, this will be the one that will go for the clinics for the human Down syndrome population to reduce DYRK1A. We still have the same effect. So treating those explants in vitro reduce the velocity of the interneurons during their migration and increase opposing time. And I have no time to, I can comment that later on if you want, but it's on the same orientation. But what the main question now was for us, what are the effects of this dosage reduction of DYRK1A in interneurons? What are the consequences on the neurobehavior? And this was done by Veronique. She demonstrated that there is a increased locomotor activities in those animals which are indeed inactivated for DYRK1A only in interneurons. You can appreciate that the locomotor activities in this test, which is an open field, a very simple test. You place the mouse in a new environment, and you let it explore. And they have much more hearing effects. So this is both seen in male and females. The timing center is not affected here, but this was not the condition for anxiety. But for anxiety, we also find that there is a lower anxiety in those individuals. When we put them into what we call an elevated plus mesh, so there are arms here which are closed, and they are open to two arms here, and the mice, they don't want to go into open arms. They are probably afraid by not being in a secure place. But still, those mice which are mutant for DYRK1A in the interneurons, they are spending more time they are doing more entries into the upper arm and spending more time also in this place. So showing a reduced anxiety level. What could be also of interest is that when we did some social tests and some learning and memory tests, and what we found is that the social novelty, for example, is abrogated in those mice. When they are inactivated for DYRK1A only in interneurons. So there's the mice which are mutants are not considering the novel congenier as a novelty compared to the other one, which are spending more time with them. And they have reduced number of contacts. And when we look for the object recognition test, so if the mice are placed with she has previously been in front of two objects. One is that will be the familiar one at the end, and there will be one which will be changed for new. Those mice which are affected here are not able to make the difference between the novel and the familiar one. So it seems that on both sides here, both on the glutamatergic and the GABAergic, this function of DYRK1A dosage is very important. But what was really also striking is that if we look now at epilepsy or sensitivity to epilepsy using PTZ, for example, or just looking at the EEGs in those mice, we can see that they are much more sensitive and prone to have clonic seizure compared to controls, and much more severe one with tonic seizures that we need to euthanize the mice at the end. But for those, when we follow only the EEG, we can see that they have much more clonic state also. So much more epilepsy is observed in those mice which are inactivated for DYRK1A only in the inhibitory neuron. So I think that if we have if you are looking for a treatment for epilepsy in DYRK1A syndrome, I think we should try to target the interneurons first, because this could be the way maybe to counterbalance the defect and to avoid the epilepsy in people. So these are my conclusion. So I try to show you that DYRK1A is implicated in the development of cortical interneurons. Glutamatergic neurons we already published. This is new story. It leads to a decreased total number of interneurons in the cortex. An alteration, this is due to alteration of their migration, and probably through the stability of actin and the contractibility of the actomyosin system, which is involved in the nucleokinesis. The kinase activity of DYRK1A is really key here, because if you don't have that, or if you destroy the kinase activity, or if you use kinase inhibitor, you are going to have the same effect. And this is crucial for behavior and cognition, and probably more important for epilepsy. What could be the next step? I have the same question as you already asked, and I think for me, we have been working a lot on trying to understand the role of DYRK1A during development and to see what could be the consequences of losing one copy of DYRK1A after birth onto the behavior and other function. But my question of now is what can we do if with a magic strand we can restore the function of DYRK1A just after birth? And see whether or not we can restore some of the functions that was not so well developed during development. Of course, the earlier we will intervene, it will be better, probably. But at the moment in human, we cannot do that. So this is something that we can do. We have the tools to do that. And I think that will be a nice avenue to pave the way for a drug of gene therapy, because you will be able to restore the functionality of DYRK1A and to see how far and how much you can restore as a function compared to any treatment that you will use. And how we can do that, we have the tools, the genetic tools. We can do conditional restoration of gene function in a disease model. This we can do in mice. In human, probably not. We cannot do that yet, but probably in the future we should be. And we have done that already for 2 types of disease in our institute, and we are doing this for the cancel-1 mutation in the Coulomb-de Vries. And we got the mice on shelf, actually, for the Coulomb-de Vries. And I will stop here, and I would like to thank especially Vicky, Véronique, and Victorine, and also Maria Delma, who was really key for another part of the project that I have no time to discuss. And the team of Juliette Godin, we are all located here, so it's quite close to Amélie, in fact. We are just 1 floor above Amélie. And that's a nice environment to work on DYRK1A, that's for sure. And thank you for your attention.
Hi, really beautiful. You were saying that the DLX56 group only turns on after they're done with liberating. Yeah, have you tried it earlier to see if the overall balance between excitatory and inhibitory is changing?
No, we haven't done that. We could have used another type of DLX3 lines, the 2 and 3 are earlier, for example. But the problem is that we were suspecting this problem of migration. And if we have impaired the proliferation of the progenitors, we would have been in a difficult position to sort the 2 systems, you know.
Oh,
so no, this is why we would like to address that using this DLX5. Of course, we can probably do that with an earlier transgene. And we didn't want also to change all the cells which are needed to support the migration of the internals. But that could be done, yeah. And there is another group who has been using an EMX2 driver, which is earlier, for example, in the glutamate technology. This is published already, so I'm not going to replicate that. But that's, of course, that could be. We have a.
Perhaps hydroxylate could be higher, and then you would actually get more interneurons born. But then maybe they do all have trouble migrating too, right? So I think it'd be interesting. I like it. Well, right?
Yeah.
Really people do.
But I don't. So we have been doing. So Victorine has been doing the same type of analysis now on Down syndrome model, and the story is it doesn't affect the proliferation too much. In fact, the dosage of DYRK1A.
Oh.
But it's still ongoing analysis. So.
I have a question: whether you see any differences in any of the assays between the kinase K2R and no. The reason, let me tell you.
No, I understand. No, I know. So I know. No, I think we should.
When you bring one allele, one from the original breeder, has to be reported to be a kinase null. It's not in the same residue. But
okay, it's another.
Negligible level. It is viable and fertile to have these flies in the lab. This strong allele is lethal. And so what's your take on that?
I agree. There is a scaffold. So for people, so protein alone is able to have some function without the kinase activity. This is for sure. And we have a few results on that. I did not have time to present that, but this is ongoing work. And we have found some slight differences between the deletion, for example, of DYRK1A, the whole deletion, and having kinase date. So there is probably what I call it a scaffold function, you know, allowing probably other partners to bind and to interact without the need of being phosphorylated by DYRK1A. No, I agree completely. I agree. I think we should discuss that, but I'm sure, yeah, this is for sure. But still, it's not, these are not easy mouse to breed because we don't have, for example, 50-50. We don't, so when you, oh, sorry, I'm taking a jargon of geneticists. So when you segregate alleles, you should have one for your father and one for your mother. So usually it's 50 of one side and 50 of the other. Here, whatever the side, whatever the mutant allele is coming from, mother or female, we only get 30% of individuals which are alive at birth. So so that means that some of the mutants carrying haploinsufficiency of DYRK1A and even the K188R are indeed dying during development for reasons that we have not investigated. So that's another level of complexity. And these are heterozygotes, not homozygotes. Homozygotes, they are not viable at all.
Yeah, actually, I've got two questions. So the first is that you mentioned sort of the gain of function sort of on the variant. And I mean, oftentimes, we see the comparison all the time between Johnson and DYRK1A syndrome. But, Johnson sort of having the 3 copies function variant. Do you see that there are distinct phenotypes
Yes.
between the gain of function and
the size, the cephalic size? In gain of function, we have a macro, a bigger skull. In the loss of function, you have a smaller, a microcephaly. I don't know. I'm pronouncing in French, so that's why I'm making it. But yeah, and that's for me quite critical, I think. Yeah. And we got the transgenic for DYRK1A is having a macrocephaly. I can show you. The rat is also the same. I can show you. We have. I didn't put that, but we have results showing that there. And this is completely different in terms of skull formation and skull differences. So you can really decide, and I would love to see that. So maybe that could be one screen to sort. I don't think it will be the only one to make, because we should be very cautious here, because you are going to treat with an inhibitor of DYRK1A. So I don't want to treat somebody who has a defective DYRK1A already with a further inhibitor of DYRK1A. Should not do that, of course. But still, I think this could be one of the features to look at first.
I guess my second question was just around it's still sort of a gain of function here. So in other disease states like hemophilia, in the development of gene therapies, they don't just use the same wild-type gene. They actually use like a gain of function variant to get an higher expression. So that everybody that's involved into sort of development therapies with gain of function variants.
No, not yet. No, I don't think so. So there are people doing that. So that's true. There are people doing that for certain myopathies. There is one group in our center, the group of Jocelyn Laporte. He has been working on a complex with 3 main actors. There are different types of. So Jeremy can talk about that too, because this and there are mutations which are loss of functions, others which are gain of function, and sometimes using a gain of function over a loss for another gene, it can rescue the phenotype. And so I don't know. I think that will be a matter of discussion. But there was a proposal which has been published a few months ago, I think, maybe a year ago now, about phosphatase, which are specific for DYRK1A. That could be one key target to look at, to decrease the level of the phosphatase that is counteracting the effect of DYRK1A as a treatment. But I haven't done that yet. We still are doing a lot of things. Yeah. Okay, thank you.